Prophase 1. At the beginning of prophase 1, the chromosomes have already duplicated. They coil to become shorter and thicker. From there, crossing over, which is the exchange of corresponding segments of DNA molecules by non-sister chromatids, occurs at the chiasmata during synapsis. The nucleolus disappears. Centrosome movement, spindle formation, and nuclear envelope breakdown also occur.
Metaphase 1. The centrioles are now at opposite poles of the cell. Pairs of homologous chromosomes are arranged on the metaphase plate with one chromosome in each pair facing each pole. Spindle fibers from one pole of the cell attach chromatids of one homolog to kinetochore microtubules. The same happens for the opposite pole.
Anaphase 1. Breakdown of proteins responsible for sister chromatid cohesion allows homologs to separate. The homologs then move toward opposite poles as a result of the action of the spindle. The sister chromatids remain attached at their centromeres and move together toward the poles.
Telophase 1. The homologous chromosome pairs complete their migration to the two poles. Now, a haploid set of chromosomes is at each pole, with each chromosome still having two chromatids. A nuclear envelope fully forms around each chromosome set. The spindle disappears and cytokinesis follows. After cytokinesis, each of the two daughter cells has a nucleus with a haploid set of replicated chromosomes.
Prophase 2. Although chromosome duplication took place prior to meiosis 1, no new chromosome replication occurs before meiosis 2. The centrioles duplicate, the nuclear envelope breaks down, and the spindle apparatus forms.
Metaphase 2. Each of the daughter cells completes the formation of a spindle apparatus. Single chromosomes align on the metaphase plate. For each chromosome, the kinetochores of the sister chromatids face the opposite poles, and each is attached to a kinetochore microtubule coming from that pole. This is so...
Anaphase 2. The centromeres separate, and the two chromatids of each chromosome move to opposite poles on the spindle. The separated chromatids are now individual chromosomes.
Telophase 2. A nuclear envelope forms around each set of chromosomes. Cytokinesis takes place, producing 4 daughter cells, each with a haploid set of unreplicated chromosomes. Because of crossing over, some chromosomes have recombined segments of the original parental chromosomes.
During prophase, the chromatin fibers become more tightly coiled and condense into visible chromosomes. Each duplicated chromosome appears as 2 identical sister chromatids joined together at their centromeres and all along their arms by cohesions. The nucleolus and the nuclear envelope disappear. The mitotic spindle forms as the centrosomes move away from each other.
Metaphase is the longest stage of mitosis. The centrosomes are now at opposite poles of the cell. The chromosomes come together on the metaphase plate. For each chromosome, the kinetochores of the sister chromatids are attached to the kinetochore microtubules coming from opposite poles.
Anaphase is the shortest stage of mitosis. When the cohesion proteins are cleaved, the sister chromatids of each pair part, resulting in 2 daughter chromosomes. They begin moving toward opposite ends of the cell as their kinetochore microtubules shorten. The cell elongates, and by the end of this stage, the 2 ends of the cell have completed collections of chromosomes.
2 daughter nuclei form in the cell. Nuclear envelopes arise from fragments of the parent cell's nuclear envelope. The nucleoli appear, and the chromosomes become less condensed. Mitosis is now complete. In cytokinesis, the division of the cytoplasm reveals 2 daughter cells. In mitosis, a parent cell can be haploid or diploid.
In meiosis, a parent cell is always diploid.
In mitosis, the number of chromosomes remains the same after division.
In meiosis, the products contain a haploid number of chromosomes.
In mitosis, the genotype of the daughter cell is identical to that of the parent cells.
In meiosis, the genotype of the daughter cells is different.